prestigious conference. First of all, I'm sorry for the inconvenience of these technical difficulties. Today, I'll be talking about recent advances in selective targeting of oncogenic forms of EGFR, and as a change or moving from organic synthesis in the last previous talks, which I really enjoyed. But my talk will be most about the design and the design strategies and molecular modeling approaches to target mutant forms of epidermal growth factor receptor. This is a brief outline of my talk. First, I'll give an introduction and background about the target, and then we will discuss the challenges in targeting mutant EGFRs and the modern strategies applied for the targeting and the molecular modeling approach. I'll discuss our publication, our recent publication at Zuhail City on EGFR modeling. And finally, if we have time, I know that we wasted some time for these technical difficulties. I'll cover the PROTACs and the combination therapy. First of all, what's epidermal growth factor receptor? Actually, it's a member of the ERB family of receptors, and it's a transmembrane tyrosine kinase receptor with the endogenous ligand of epidermal growth factor and EGF and TGF alpha. And as you must know, small molecule tyrosine kinase inhibitors are now widely used for treatment of various types of cancer since the breakthrough discovery back in 2001 of imatinib, which is an ABL-BCR inhibitor. And before that date, the idea of using inhibitors for tyrosine kinase looks not applicable until the advent of this breakthrough drug, imatinib or Gleevec, which is approved by FDA for treatment of leukemia in 2001. And as we know, EGFR acts as a dimer, and it's a transmembrane tyrosine kinase, and this is a schematic at the bottom of the slide. It's a schematic representation of the structure. It has an extracellular part followed by a helical or mostly helical transmembrane domain, and then the kinase domain is actually the intracellular part followed by some cytoplasmic end. This is a more clear representation of the tyrosine kinase. We have 4 extracellular domains. The biggest one is the one on red, figure B, followed by the transmembrane helical part, and finally the tyrosine kinase part, which is of interest to us in blue color. Epidermal growth factor is a key player in various types of cancer, especially lung cancer and breast cancer. It stimulating or phosphorylating a lot of downstream effector molecules and kinases by phosphorylating these kinases and initiating the signaling cascade mainly in the MAPK, AKT, and GNK kinase pathways. And this eventually leads to stimulation of cell proliferation, inhibition of apoptosis, angiogenesis, and migration, adhesion, and invasion, and finally to metastasis of some important or stronger types of cancer. First of all, I don't know if you are interested in molecular biology or molecular mechanisms of certain enzymes, but it's an essential part and the cornerstone to understand the strategy of drug design. These are 3D structures or X-ray crystal structures of the insulin kinase receptor. This is the inactive form and this is the active form. Kinase domains generally are composed of 2 lobes, the N-terminal lobe and C lobe. And of course the hinge region where the ATP binds. This is the inactive form where the activation segment is closed. And here this is the active form of the kinase. We can see that the activation segment in red is open. And opening the ATP binding site, this is the ATP, or actually it's a modified ATP. And this is the substrate binding site, the substrate protein. It can be another kinase, of course, or or it can be actually the activation segment of another kinase, which is being phosphorylated by this kinase here, the blue one, this is substrate. I'd like to give you a brief introduction about, or reminder about the molecular mechanism of tyrosine kinase. This is a tyrosine residue which is being phosphorylated. We have two critical aspartic acid. The second one here, or the lower one here, forms a charged as tested hydrogen bond with the OH of the tyrosine, which increases its nucleophilicity to attack the phosphate atom. It's a nucleophilic attack on the gamma phosphate of the ATP. And again, it's also facilitated by coordination with two magnesium ions and the first aspartic acid here. So it acts like a small reactor, like we do in the lab, but it's a natural small reactor. And this is the active form, of course, and in active conformation, we don't have space for the binding of ATP or even the other protein substrate. And of course, we have two designation for this. We call the active form the alpha CN, the active form to the right, alpha CN, because we have the alpha helix here move inwards, and we call it DFGN also. So the active is alpha CN or DFGN, and the inactive is alpha C out or DFG out. So the EGFR inhibitors are proved or were proved effective in treatment of various types of cancer. These are the first generation EGFR inhibitors approved in 2004 and 2. It's gefitinib and erlotinib, and they are active against sensitizing mutation. Excuse me. L858R and exon 19 deletion. So what are these sensitizing mutation? Here, this is the natural mechanism. This is the inactive, and this is the active form, where the alpha C helix move inwards upon activation. And we have, of course, two types of inhibitors, as you know, type 1 inhibitors, which binds to the active form, and type 2, which binds to the inactive form. Upon this mutation or exon 19 deletion, the loop connecting the beta 3 helix and the alpha C getting shorter upon this mutation, and the kinase or the equilibrium is now shifted towards the active form of the kinase, which allows the binding of only type 1 inhibitor, and type 2 inhibitors are no longer finding a place for the binding, because the pocket is getting smaller because of this constitutive activity of the kinase. Also, the other sensitizing or activating mutation making this kinase oncogenic one is the L858R mutation. It's leucine 858 is a hydrophobic residue, amino acid residue, as you know. It's located in the activation segment, and it lies in the place between beta 3 and alpha C helix, where it's hydrophobic, making hydrophobic interaction with these residues. And once it's mutated to arginine, it loses this hydrophobic interaction, and this hydrophobic interaction are no longer possible, and this forcing the activation segment to the open conformation or the open state, and again the enzyme is now constitutively active. But anyway, first generation EGFR inhibitors are active against all these sensitizing mutation, because it binds to the active form or the active conformation of the protein. Now we have another problem. The gatekeeper methionine, or the gatekeeper threonine 790, which is relatively smaller, is now mutated after around 1 year of treatment to methionine 790, which is bulkier, as you know. And this gatekeeper residue is able to hinder the binding or the access of the first generation inhibitor to the back pocket, to the hydrophobic pocket of the EGFR. So scientists developed the second generation of EGFR inhibitors, where they have an electrophilic group, it's acrylamide derivative, to form a covalent, stable, irreversible covalent bond with cysteine 797 at the perimeter or at the edge of the ATP binding site. So we can understand what happened from this figure. This is serine 790, which is involved in a water-mediated hydrogen bonding with the nucleus of the inhibitor. But now when it is mutated to the red one, to the pink one, methionine 790, which is larger than threonine, so we can see the steric clashes which hinder the entry or the binding of the inhibitors to the ATP binding site. So this competition occurs, and the enzyme is now has more affinity to the ATP, and this reduces the affinity to our inhibitor. So second generation, which binds covalently to the enzyme, solved this problem, but we have another problem. We have limited clinical use because second generation EGFR inhibitors are not selective against the mutant form. They inhibit also the wild type EGFR, causing a lot of toxicity like skin rash and GI toxicity because it's non-selective and it inhibits all types of EGFR, not only the oncogenic forms. So we had to develop the third generation EGFR inhibitors. It's very similar and use the same strategy as the second generation EGFR inhibitors, but now we have a slightly compact shape and the size of the receptor of the inhibitor, and this inhibitor can adopt a U-shaped conformation, avoiding the steric clashes with methionine 790 and actually making favorable hydrophobic contacts with it. Actually, this conformation is natural to the bioactive conformation of the ligands. It has a pyrimidine core with a U-shaped structure, and from these figures we can see that this osimertinib derivatives, the prototype was osimertinib, and then there are a lot of derivatives developed later on. We can see the selectivity against the wild type of or wild type form of EGFR is in the range of hundreds, 200 to 400 or 600 selectivity against the wild type. So it's more selective against the oncogenic, or more selective for the oncogenic forms of the protein. At Zuhail City, we published last year in 2022 a study on the structural basis for the selectivity of third generation inhibitors, which could help scientists to develop more successful selective inhibitors for the oncogenic EGFR. We performed 300 nanosecond MED simulation. This is the crystal structure of the wild type bound to osimertinib, and this is a double mutant with the osimertinib inhibitor bound in the same active site, the ATP binding site. And we can see that here methionine 790 forms hydrophobic contact with the pyrimidine core. At the end of our study, we concluded that the selectivity for the mutant form over the wild type form can be attributed to two factors. One is a direct factor and the other is an indirect factor. The direct factor is the hydrophobic, strong and stable hydrophobic contact, van der Waals contact between the pyrimidine core of the osimertinib and methionine 790. Here we can see the wild type complex and the double mutant complex before and after the MED simulation. The initial poses are in gray in both figures, and the poses after the MED simulation are in green and cyan. We can see that there is a little shift here in double mutant of the pyrimidine core or the osimertinib towards the methionine 790, so they are getting closer, making favorable hydrophobic contacts. And also this hydrophobic contacts push the ligand slightly outwards, which making the hydrogen bond with the hinge region stronger with methionine 793, which is essential for kinase inhibition. And also it strengthens the hydrophobic interaction with valine and leucine. And most importantly, it brings the Michael acceptor or the acrylamide group closer to the cysteine to form the covalent bond more easily. The indirect reason is it's more active against the sensitizing mutation L858R, because the arginine now in the open conformation can make a hydrogen bond with tyrosine 891, fixing the enzyme in the active conformation or the active form, which is suitable for the binding of osimertinib. Osimertinib is still a type 1 inhibitor. It binds to the active form, and this sensitizing mutation holds or stabilizing the EGFR in the active form suitable for the binding of osimertinib. This was the conclusion of our study, and this is the U-shaped conformation of osimertinib. But finally, we still have another problem. Now, after 10 months in the clinical trial of osimertinib, after 10 months of treatment with osimertinib, 50% of patients unfortunately develop resistance to osimertinib, again, due to mutation of cysteine 797, the target of alkylation of this alkylating agent, to serine, which is less nucleophilic, because we have OH group rather than SH group here. So this mutation renders the third generation tyrosine kinase inhibitors ineffective, again, due to the less nucleophilic serine. Now the problem is, the current problem is, or the gap in science is, we have first and second generation inhibitors for activating mutation deletion 19 and L858R. And for patients who are having T790M mutation, we can treat them using osimertinib. This is some kind of precision medicine. We don't move to osimertinib unless we try erlotinib. But again, some patients develop resistance to osimertinib due to mutation of cysteine 797 to serine, which is the last problem that we don't have a solution so far, or at least a solution in, maybe we have some compounds in clinical trials, but we don't have an FDA approved solution for the last case. So what are the solutions? We can target serine 797 instead of cysteine, or design allosteric inhibitor and abandon the ATP binding site altogether. Briefly, I'll go quickly to meet the time. We can shift our interest to serine 797 by installing hydrogen bonding groups to bind with serine and the nearby aspartic instead of cysteine. We don't need the acrylamide anymore, so we can use just hydrogen bonding groups. And also we can target other polar groups by hydrogen bond or ionic bond on the premises, on the perimeter of the ATP binding site, and leave the cysteine alone or the serine alone. But again, these compounds lost the selectivity against the wild type. The other solution is developing allosteric inhibitor. We found a small hydrophobic back pocket in the ATP, not our group, of course. It's published by another group 2 years ago. And the prototype compound was EAI045, and follow-up derivatives are JBG with some code, as you can see here. These are allosteric inhibitors bound inside the hydrophobic pocket besides the ATP. This is adenosine monophosphate, so it's not competitive with the ATP. It binds as a 3-blade propeller here. But again, it still have a problem because, as we know, EGFR is acting as a dimer, and it cannot bind to the receiver unit. We have activator and the receiver unit. It binds to hydrophobic back pocket, as you know, but the allosteric site is not accessible in the receiver unit, in this unit. And so it's still active, producing the oncogenic effect. So it binds only to one unit, and we have to use it in a combination with cetuximab, which is a monoclonal antibody that inhibits the dimerization of EGFR. So it's very successful if used with cetuximab, but it's not active alone. So is the problem solved? Unfortunately, it's still not solved because cetuximab is not selective, again, against the wild type. It inhibits both forms, causing toxicity. So this is a gap of knowledge or a gap of science. This is a problem that scientists are working on it now. And there is another completely different approach, which is PROTACs, which is proteolysis targeting chimeras. These are trimeric small molecule composed of selective modulator for the target protein or inhibitor of EGFR, linked to a ligase recruiting ligand with a stable linker. Ligase enzyme, as you know, install ubiquitin units or ubiquitinate their target proteins and label these proteins for degradation by the proteasome system. So once bound in this way, this is the EGFR ligand, and this is the linker, and this is the E3 ligase ligand. Once bound in this way, the E3 ligase ubiquitinate the EGFR or whatever the target protein causing degradation. And I'll skip this optoprotac. Protac showed superior potency and specificity compared to traditional small molecules drugs, and these chimeras also possess a longer duration of action and could overcome the drug resistance problem. This is the design strategy of EGFR Protac. This is osimertinib, and this is the ligand for E3 ligase, lenalidomide, and we install linker on this NH2 and this N-methyl group of osimertinib. This is the first EGFR Protac, osimertinib Protac, with good potency, submicromolar potency against different cell lines. And Protacs are developed for many kinase inhibitors. And this is a list. This is a list of the reported EGFR Protac inhibitors. The ligands in pink are EGFR ligands. The linker is in black, and the E3 ligase is in blue. Finally, I'm editing a research topic in Frontiers in Drug Discovery on Protac, so you are welcome to submit your articles, whatever they are, original research articles or review articles. We have an abstract submission deadline by the end of March and a manuscript submission deadline by the end of July. And this is the conclusion, because we don't have time. Of course, you can see these points and you didn't forget what I have said. And thank you for your time and patience.